Great. So this is actually going to be a little different because I want to actually sort of make the case for basic science here. And I know that, in the introduction, we were talking about, the vision of the foundation is, the next 3 to 5 years. And a lot of what we've been talking about is in human studies, and that's, absolutely critical because that's what we care about curing. We don't care about curing the mice. But I do think that particularly when we're talking about repurposing drugs that are already in the pipeline for other things, basic science and mouse science is a good source of identifying new targets that we might not have considered and sort of new pathways, new biology that we might not have considered that I think can be very informative even in the 3 to 5 year mission of trying to advance new drugs through the pipeline. So we're going to hear a little bit of mouse science now. All right, so we've beaten these guys into the ground so far this morning. The IL-1 family members are obviously very attractive targets in MAS, in part because of the relationship to systemic JA, and there's certainly good clinical data in a number of different scenarios. We've heard a lot about IL-1 and IL-18. I'm going to actually talk to you guys about IL-33, which is yet another member of this family. These guys do a lot of different things. This is just, again, one sort of simple slide to show one of their major functions, which is directing the other types of cytokines that different T cells make. So IL-1 will direct CD4 T cells to make IL-17. IL-18 will direct both CD8s and CD4 T cells to make interferon gamma. And IL-33 has been studied for like the last 15 years, maybe 20 years, for its effects on CD4 cells in directing them to make IL-4, which is a cytokine that's really important for allergy and asthma and those kinds of things. So actually there's IL-33 drugs in the pipeline in a couple of pharmaceutical companies with the intention of trying to treat things like asthma and eczema and allergy. But more recently, there has been attention for the role of IL-33 in directing CD8 T cells and making interferon gamma. And we've talked a lot about interferon gamma today as being an important target for these diseases. And that sort of raised the question in our mind whether IL-33 couldn't be repurposed for the sake of things in the MAS or HLH category. So I'm going to try and move quickly through the slides, because we're, want to stay on time. So I'm going to start with some published data that I'm going to try and move through very quickly. I'm going to make the long story as short as I can, which is that you're just going to have to trust me. We had good reason to do this. We decided to look at IL-33 blockade in mice that get primary HLH. So these are perforin deficient mice. You infect them with a virus. They get a syndrome that looks just like primary HLH, and they die. And the blue line is what happens to mice if you give them nothing, basically, or you give them a control treatment. And over the course of about 18 days, about 80% of the mice will die of HLH. And if you block IL-33 by giving a monoclonal antibody that blocks the IL-33 receptor, in this experiment we got 100% survival. Sometimes one mouse might die, but it was a very dramatic difference in survival when we block IL-33. And again, trying to make the long story short, this seems to work, we think, because what it's doing is it's blocking the amount of interferon gamma that's being made in these mice. So we get a 16-fold reduction in the interferon gamma in the blood of these mice. And that amount of interferon gamma in that red bar that you're seeing, which is the IL-33 blocked mice, that's the amount of interferon gamma that a normal wild-type mouse would make if you infected it with a virus. So what you're doing is you're detuning the amount of interferon gamma, not to zero, but to sort of normal levels. Again, so for the sake of time, I'm not going to spend a ton of time on this slide, but we can show that this is happening in a cell-intrinsic manner. So what that means is that this is IL-33 binding to the IL-33 receptor on CD8 T cells and blocking CD8 T cell interferon gamma production. So this is a mixed bone marrow chimera experiment, and basically in the gray bars, you're looking at bone marrow cells that come from a mouse that's perforin and IL-33 receptor deficient. The black bars is bone marrow from just a perforin deficient mouse, and then you make chimeric mice, either mice that are 100% perforin deficient, 100% doubly deficient, or a 1-to-1 mix. And what you can see is that in the mixed mouse, the cells behave like the cells if they were in a completely full mouse, meaning that the effect of IL-33 or ST2 blockade or interferon gamma, IL-33 receptor blockade is acting directly on the cells to block that interferon gamma production. So that was really exciting. We have this mouse model. IL33 blockade seems like a kind of a nice target for the treatment of primary HLH, at least in this mouse model. It seems to work through a mechanism that makes sense with what we already know about these animals, which is that interferon gamma is really important. What about humans? And so we went and looked at data that's a combination of data from both Alexei Gram and from Michael Jordan, where they've done gene transcription profiling from peripheral blood cells from patients with FHL. And we're showing you the 3 patients that had perforin deficient FHL, systemic JA, and healthy controls. And what you can see is that specifically in the FHL population, there was an upregulation of the IL33 receptor in the blood of those patients, suggesting that it might in fact be a target. So this led us to this sort of new model, and again, I'm just going to go quickly because this has all sort of been published, whereby there's an interferon gamma axis and an IL-33 axis. They're sort of integrated with each other, whereby IL-33 controls both the number of CD8 T cells present, the amount of interferon gamma that they're producing, and together contribute to disease pathogenesis. And so, that suggests that IL-33 might be another interesting target for these patients. Certainly, the interferon gamma trial is, already ongoing, and that's likely to be, a viable target as well. And there are some advantages and disadvantages of each treatment paradigm. But I think, the more kind of targets we have, the more chance we're going to be able to cover all the population. So, I'm not a clinical trialist either, and I think now the sort of challenges are getting pharma that already have this drug in their pipelines that they're not necessarily moving forward on for allergy and atopy because maybe it's not the best drug for that. Is there interest in repurposing it for FHL-type pathophysiology? But one of the other things that we can do, again, in the mouse world, is we can ask about sort of the robustness of the target. So ideally, you'd like the drug to operate under a large array of circumstances. And because, as has been said before, humans are not inbred mice. You do one model, you've done one model. But what you'd like to do is know that this thing works no matter how you tweak the system or what else you throw at it. And so, we can do that in mouse models. We can tweak the model and ask, well, do things still operate the way that we'd like them to? So one tweak we did was to ask the question, what would happen if these mice already had a pre-existing memory response to the virus, right? Because human beings living out in the real world are not mice living in a specific pathogen-free colony. They experience other viruses. There can be T-cell cross-reactivity. There are former FRUSTA FHL patients that, don't present until later, and so maybe they've already had a first infection and now have pre-existing memory to subsequent infections. What happens if you immunize mice to the LCMV, perforin-deficient mice to the LCMV virus, and then give them LCMV? Do they get a worse FHL? Do they get a different FHL? Is it still responsive to these therapies? So again, I'm going to try and summarize these data really quickly. I've already presented them in other meetings, and hopefully we'll have this paper out for review in a couple of weeks. So if you immunize mice against the LCMV virus, and it doesn't matter what epitope you immunize them with, you can use different epitopes, they all get an enhanced disease. So they die faster. I'm not going to show you the data, but they get worse CBCs, they get higher ferritins, they get more liver inflammation, they lose weight faster, and so there's an enhanced disease. And then you can ask the question, is this enhanced disease going to behave to the drugs the same way as just regular vanilla FHL? I wouldn't be showing these slides if the answer was no, so of course there are some differences. So this is a really complicated slide. I'm going to try and go through it real quickly. Let's see, can I get a pointer up? Yes. All right, so the blue line is an unimmunized mouse that has not been blocked, has not received any drug. So this is your vanilla FHL, and that's what I showed you before. If you do nothing to the mice, they die. The red line is a mouse that's been immunized. So as I just showed you before, if you immunize the mouse, they get an enhanced disease, they die faster. Okay? The black line is an unimmunized mouse that's been blocked with interferon gamma, and that's what I've told you before. If you block with interferon gamma, the mice do much, much better. And that's been shown in many different versions of FHL and time again, and we can reproduce that here. But what was interesting is that if you take the green line, which is the immunized mouse, and now block with interferon gamma, the mice do a little bit better, but they don't do anywhere near as better as mice that have not been immunized, right? And so you tweak the system, and now the drug isn't working quite as well. In fact, the green line looks a lot like the blue line, which is the unimmunized mice, no treatment. And so now, for whatever reason, in this system, interferon gamma blockade is not as efficacious as we would like. We've done all the controls to show that, we're getting the right pharmacodynamics, we're dosing the drug correctly, we are blocking interferon gamma, it's just not saving the mice. And the orange line is mice that are now getting both interferon gamma and TNF alpha blockade. And those mice now start to do better. They look more like the black line. So what has happened is that a new cytokine is now playing a new role, TNF alpha, and it's actually the combination of both cytokines that are making these mice sick. Again, I'm skipping a lot of data. The TNF alpha is coming from the CD8 T cells. We've sort of done all the science to show where it's coming from. We just don't have time to get it into today. So again, I didn't want to make the slide too crazy. If you block TNF alpha alone, it gives you no effect. And then if you do all the combinations in unimmunized mice, it actually has no effect. And that's been shown by Mike Jordan years ago that TNF alpha does nothing in the vanilla model. We can reproduce that. And it looks just like the red line if you do it here. Singly, it doesn't do it. You have to have both. I'm just going to skip that part for a second because I don't have time. So, I'm sorry, oh, that's all right.
What else did you try to block in this model besides?
We tried to block IL-6, I think we did. I think just IL-6, and we went quickly to TNF alpha because we had the data that we saw increased TNF alpha levels both in the serum and being produced by the CD8 T cells. But the real reason why I'm showing you the data is this slide, right? Which is that if you go upstream, if you block IL33, it still works. And it still works probably because you're blocking now, just by blocking the upstream signal of IL33, you're blocking both the TNF alpha and the interferon gamma all in one pop by blocking a receptor that's controlling the levels of both of them. And we can show, again, if you block the IL33, the interferon gamma and the TNF alpha both come down in these mice, right? So at least in this setting, for what it's worth, it's a contrived mouse model. I'm not saying that somehow, human beings have been immunized with dendritic cells pulsed with peptide to a virus and that's, how they're getting their disease. But in a setting, IL33 is showing a benefit that maybe interferon gamma blockade does not. And so that might be an attractive thing about using this approach. What about another tweak? And I'm going to be sort of purposely provocative here. And again, I don't have time to show you all the data, but I'm happy to talk about it with anyone who wants to talk about it afterwards. So we got interested in these reports and then there are two of them, case reports of families that have an interferon gamma receptor deficiency, and yet there are patients from these families that develop what looks like bona fide HLH. And how the heck does that work if interferon gamma is so central to the disease? Which, by the way, I totally believe, right? Interferon gamma is absolutely central to the disease for these patients. And yet there are patients that are missing the receptor and develop what looks like an HLH-like syndrome. And then somebody had mentioned earlier on the putative systemic J mouse model, which is basically an interferon gamma knockout mouse that's been injected with complete Freund's adjuvant. And the authors sort of claim that this looks a lot like systemic JA, and this is maybe a model for that disease. And I had a really hard time wrapping my head around that model because again, the problem with mouse models is sometimes they just are a little contrived, and I'll be the first one to admit that about the TLR9 model. So this was another one that sort of bothered me, and I couldn't really figure it out. But I'm going to show you some data that maybe makes it make some sense. So because of this, we decided to do something that was maybe a little bit crazy, which was to breed a mouse that is both interferon gamma deficient and perforin deficient. And the reason why we did this is that one possibility for the interferon gamma receptor observation is that interferon gamma can bind to something else besides the canonical receptor. So if that's the case, then if we knock out interferon gamma itself, then those mice shouldn't get the phenotype if it's just about a different receptor. But if really interferon gamma can be dispensable, then we might see an interferon gamma perforin deficient mouse get sick as well. So we bred the double mice, we infected them with LCMV, we compared them to singly deficient mice. And again, just summarizing a lot of data, I'm not going to show it all to you, but we've done ferritins and soluble CD25 and CBCs, the whole shebang. I'm just going to show you survival and weight. These double knockout mice get just as sick, and if anything, in many instances, more sick than the single knockout mouse. So indeed, you can actually get what for all the world looks like an HLH-like syndrome in the complete absence of interferon gamma. the one thing that's a little bit wacky and different about these mice is that when we ran their CBCs, it actually broke our CBC machine. And it broke our CBC machine because the white counts were stupid high. And they were so high because they had a massive neutrophilia. So these double knockout mice, although they got anemia and thrombocytopenia, just like an HLH patient would, they had a really super high robust neutrophil count, like 80,000 per microliter. And that was accompanied by an increase in GM-CSF in the serum. And of course, the double knockout mice don't make interferon gamma. The perfect knockout mice make a crazy amount of interferon gamma. And so they get an HLH, but they get an HLH associated with a really high neutrophilia. And that kind of, it reminds me of that systemic JA thing, right? I mean, that's something we see in our systemic JA patients. And it kind of took me back to that Mathis model of, these interferon gamma knockouts getting this neutrophilic systemic JA-like disease. So again, just to show you, blocking interferon gamma works, right? So once again, if I just use an interferon gamma blocking antibody in perforin deficient mice, they're cured. I'm not debating that. It's absolutely an appropriate therapy. That is absolutely correct. And I actually think what these double knockout mice are is just another tweak, an alternative model. Again, it's not that I think that there are people out there walking around that are doubly deficient in the interferon gamma receptor and perforin. But when we do that in a mouse, we are replicating a different type of inflammatory milieu that gives us a different HLH-like version that maybe has something to do more with the neutrophilic MAS of systemic JA. And so just to show you, we block with the antibody, the mice do indeed get cured. And what I think the difference is that if you use MHC class 2 as a readout of the pharmacodynamics of the antibody, how well is blockade working? This is a perforin knockout mouse getting an isotype antibody. It has lots of class 2 upregulation on its inflammatory monocytes. If you block with the dose of antibody that cured the mice, you get a partial loss of class 2. So the antibody's working, it's suppressing that interferon gamma responsive gene, but it's not complete. In the double knockout, it goes completely away. And so I think that that's what the difference is. It's the complete loss of interferon gamma that sets up this other weird immunopathology. And this is just summarized over a bunch of mice here. Okay. It is still a CD8-dependent disease. If I delete the CD8 T cells with a depleting antibody, I can cure the mice, I can cure them of their survival, I can cure them of their weight loss, and I'm not going to show it to you, but all the other parameters, okay? And what I think is going on, we haven't done the final experiments yet to really nail this, but what I think is going on is that these doubly deficient CD8s have acquired a new function. They are making lots of GM-CSF. And so I'm just showing that to you in the flow cytometry here, and this is summary data. Again, I don't have time to show you all the data, but what I can tell you is that this is happening in both a interferon gamma receptor intrinsic and extrinsic role on the CD8 T cells, so it's really complicated. We've done both in vivo mixed bone marrow chimera experiments, we've done in vitro mixing experiments. I just don't have time to show you all the data. We really tried to work out the science here. But what I believe is that there is a circuit by which interferon gamma suppresses the capacity for CD8 T cells to make GM-CSF. If I go back to Mike Jordan's data set, his microarray data set, and you actually ask about those patients and their PBMCs, actually there is a cluster of patients in that data set that are making GM-CSF, both perforin deficient and the non-perforin deficient ones. What I don't have is, because the graduate student sent me the slide too late, is if actually you plot it against interferon gamma, there are all different subsets. There's one patient that's a double producer, and then there's a whole cluster that's a single GM-CSF producer, and another cluster that's an interferon gamma single producer, and then there are actually a bunch of double negatives, because as we pointed out, most of the interferon gamma response is in the tissues. And then again, why is this important for an IL33 talk? Well, if you treat with ST2 blockade, you can fix these mice, these doubly deficient mice as well. So they are still IL33 dependent. And so just in my last minute and a half, if I'm going to be provocative, we were talking about the lung disease, right? And somebody mentioned Bruce Trapnell, right? And this, there's a nice body of literature on patients that get pulmonary alveolar proteinosis as a result of as a result of GM-CSF neutralizing antibodies. And so the thought strikes me is that what our connection here is in our systemic JA MAS lung phenotype, is that when they get into that interferon gamma high physiology, that interferon gamma is now shutting off GM-CSF, and so it's equivalent to sort of being GM-CSF neutralized, and so those patients begin to develop a lung phenotype. And so maybe it's wild total speculation, okay? So feel free to, shoot me down. Could be totally wrong. But I find this idea provocative. And I think that's the power of fooling around with the mice, is that maybe there is a connection there that I would have never thought of. But that, in fact, there is a relationship between gamma and GM-CSF, and maybe that's an explanation for the lungs. So I'm just going to finish up with the acknowledgement slide. Julia Rude was the graduate student that did the initial ST2 IL33 blocking studies. The middle guy is a PICU fellow that did the immunization studies. Tom Byrne is a current PhD student in a lab who did those GM-CSF studies, and I think I have, 3 seconds for questions.
You have 5 minutes for questions.
So,
Who's less frequent than you.
oh.
Okay. Thanks for the beautiful mouse model. I just wonder that talking about the target interferon gamma, is it the NK cells the major source in the innate immunity setting, where the T cells are the major source in the adaptive setting? So if that's the case, can we exclude the possibility of the innate immunity part, for the human conditions? If that's the case, I mean, targeting on IL-23, I'm not aware whether the receptor for IL-23 is expressed predominantly on NK cells.
So right, so I think most of the IL-33 receptor is on CD8 T cells. So in the mouse model, that's been done, right? So the source of the pathogenic interferon gamma in the mouse model is the CD8 T cell. So we know that now. Again, you can't do all the crazy things to humans that we can do in mice to prove that, but I think it's likely to be the same that the pathogenic source, or the source of these pathogenic cytokines, is the CD8 T cell, which makes IL-33 a reasonable thing to think about because those cells do express it. Go ahead, Canna.
Quick question. So first, maybe I should know this, but I don't. Any other ligands for ST2?
Not that I know of.
Okay. And second, do you have a proposed mechanism by which gamma is preventing GM-CSF production?
It's no, because it's super complicated, right? So there are both CD8 intrinsic and extrinsic effects. And so, one of our thoughts is, at least for the intrinsic effects, is there sort of a chromatin remodeling accessibility issue, that these cells are sort of poised so that they can do it, but it's shut down when they get the gamma signal. There's a lot more work to be done to try and figure that out. It's going to be super complicated because unfortunately there are intrinsic and extrinsic effects. We got the very disappointing result that or maybe it's a good result, because maybe I can write a lot of grants, but we got the hard result, which is that there's not going to be a simple answer.
I also have some information questions. So do CD4 T cells express the IL-33 receptor?
Yes.
And so does interferon have any effect on their production of GM-CSF?
Not that we've measured in this system.
But you've measured it and they don't make it in this system?
Correct.
Okay.
So in your double knockouts, what do the CD8s make that make the mice sick? It is not gamma.
Yeah. So I mean, I think it's GM-CSF, but so
GM-CSF is the one that drives the.
I don't know. So we just got our order of the GM-CSF neutralizing antibody from BioXcel. So just the timing of this meeting is a little bit too early for me to answer that question. But we're really trying to answer that question. And it might not be GM-CSF, right? I mean, the GM-CSF might be just the neutrophilia piece, and it might be something totally different. So I don't know. We intend to find out. Randy.
So if it is GM-CSF and the lung disease, if you're right, you would argue that the kids who get lung disease are the kids who are just really hard to treat, as opposed to blaming it on biologics?
I think it is. I'm well, boy, you really stuck me into the controversial one, right? I am not yet convinced it's the biologics, but that may be more emotion than science. But what I would say is, if I'm right, then that would suggest it's not the biologics, but rather it's the ones that are making a over-exuberant interferon gamma response, and it's suppressing GM-CSF, and then that's what's causing it. And that is a data-free assertion, right? I've done nothing to, and I don't know that we've actually, I know that we have not looked at lung disease in these mice, right? So I've got no data. I'm just throwing a bomb out there because I want to be provocative.
Just write the IRB and look at their lungs.
We've got time for one more. Do you want to flip for it?
So I can help you. I think it's unlikely that the interferon, inhibition of the GM-CSF pathway is going to be the sole explanation of the lung disease. It may be contributing to the lung disease, but the lung pathology we're seeing is much more complex and much more inflammatory than the more simple, although simple is probably not the right word to use, form that we see in the patients with the GM-CSF autoantibody lung disease. So that may be contributing, but I wouldn't expect that to be the entire answer.
I think that's always the answer, right? Like life is complicated, but we'll work on it.
Okay, thank you. Betsy.